Hello everyone, welcome to IGCSE Fashion. Sorry for not filming, I was busy during the last month, but for the day we'll be explaining Chapter 5 Enzymes from 0610 Biology IGCSE for the extended version. So here are the points that we'll be covering in this video. We're going to cover both core and supplement. Moving on, we're going to start with our very first definition. What is a catalyst? A catalyst is a substance that increases the rate of reaction without getting used up. Now, these 3 points are required to get the full mark when it comes for definitions. So please take into consideration each and every point that is placed in this slide. Moving on, I'm going to use my highlighter as usual. So what's an enzyme? Enzyme is a protein. It's a biological catalyst and it reduces the activation energy. The activation energy is the energy required to start a reaction, so it reduces the amount of the minimum amount of energy that is required to start a reaction so that the reaction can take less time, and it increases the rate of the reaction, and most importantly, let's change the color to red, it wouldn't get used up while the metabolic reactions are going on. So it's required in metabolic reactions and it wouldn't get used up when the reaction takes place. Why are enzymes important? So what's the role of an enzyme in our life? They control the reaction in cells. They make sure that these reactions occur quickly enough for the cell to function. If the reactions were slow, because metabolic reactions may require years to take place, but with enzymes it requires seconds to take place. So if we got used to do this reaction, we may die in few days, if not hours. Now the actions that would happen. The blue part is known as an enzyme and the red part is the substrate. Now, there is no certain color for substrates and enzyme, but I'm just trying to illustrate it for you all. So, how does the enzyme and the substrate connect together to make a reaction? So, enzymes have an active site. What's an active site? This one, this part here, this area is known as the active site. It is a complementary shape specific for a substrate, and it is responsible for creating an enzyme substrate complex. You need to memorize this point, enzyme substrate complex. I'm going to put brackets around it because it's important. And the red one is the substrate. Now, when you connect the substrate with the enzyme through the active site, you're making an enzyme substrate complex. It's a lock and key relationship because they are complementary. Now, after the reaction takes place, we're gonna have a product. Now, the product would form and the active site would stay the same, okay? It wouldn't change. We said that earlier. Now, we have two types of reaction that would happen in metabolic reactions. We have the anabolism and the catabolism. What's the difference? Anabolism is connecting particles together or connecting two substrates together using energy, and we have the catabolism, which is breaking off molecules and producing energy. This one is endothermic, this one is exothermic. Enzymes and temperature. Now, enzymes may get affected by two things. Number one is temperature, number two is pH. We're going to start with the temperature. So, what does the increase of temperature have an impact on the enzyme? As the temperature increases, the rate of the reaction will increase in most chemical reactions. When the temperature falls, then the reaction, like the rate of the reaction, will slow down. Now, usually a rise of 10 Celsius will double the rate of an enzyme-controlled reaction in the cell, but the optimum temperature is about 37 Celsius. Now, 37 Celsius is like the body temperature, that's why it is the optimum temperature. Optimum temperature is the highest temperature before an enzyme would denaturate or would no longer work. This is because the enzyme substrate molecules are constantly moving using kinetic energy. So, like they're not stable in place, like the substrate would be moving around the cell as well as the enzyme. So, they are in constant motion, and when the temperature increases, the kinetic energy of both the enzyme and the substrate will increase, and so it will have a higher rate of reaction and a higher probability of making a complex. The reaction will only happen when the enzyme and substrate molecule collide with each other. Like it wouldn't take place when the enzyme is on side and the substrate is on the other side. As the temperature is increased, the molecule gain kinetic energy. Here are the keywords that you need to talk about when they tell you that the temperature increased. Firstly, the molecule gains kinetic energy, then they move faster, higher chances, then a greater chance of collision. Here's number three. So the rate of reaction would increase. Now I'm going to explain more of what would happen if the temperature increases moving on. So above the optimum temperature, which is about 50 Celsius, you don't need to know that. Sometimes like it would depend according to the type of enzyme and the structure and other, let's say, effects. Now, above the optimum temperature, which is, as we said earlier, the optimum temperature is the highest temperature before denaturation. The molecule gain even more kinetic energy. Here is point number five. But their action will slow down. This is number six. This is because enzyme protein molecules start to lose their shape, which is known as denaturation at a higher temperature. So the shape of the active site would change. This is 8. So they will no longer be complementary. So although there are more collisions, here's number 9, due to the molecules having greater kinetic energy, then the number of effective collisions would reduce. This is 10. Substrate molecule cannot fit together with the enzyme, stopping the reaction. That's why I'm going to explain on this graph at the end. Denaturation is a permanent change. This is very important. When temperature increases and it would bypass the optimum temperature, we will have a permanent change. What do we mean by that? Even when the temperature is reduced below 37 Celsius, it would never go back as it was and the reaction will no longer take place. So going to this graph, the one that I'm going to be explaining on, I'm going to just repeat the points that I said earlier. As temperature increases, the rate of reaction increases, the collision rate will increase at a certain point, which is the optimum temperature. It will have the highest rate of reaction and the highest rate of collisions. And when temperature increases even more, the rate of reaction will decrease and effective collision will reduce because enzymes will lose its active site shape due to denaturation. And the reaction will stop at, let's say, 60 Celsius if it was 60 Celsius. And it will be a permanent change. Moving on. Now going to enzyme and pH, acidity or alkaline conditions can denaturate some enzymes. So most enzymes work at a particular level of acidity. Now the difference between an enzyme with a pH, different pHs and different temperatures in temperature, most enzymes would have optimum temperature of 37, which is the body temperature, while pH would have different pHs depending on the type of enzyme and the location of the enzyme. So protein digesting enzymes in your stomach, for example, will work in an acidic condition, which is pH of 2, which eventually the amylase, which is found in the saliva, would never work at. Inside the cell, most enzymes will work best at a neutral condition, which is pH of 7. The pH or temperature at which an enzyme works best is called optimum pH or optimum temperature. Conditions in the duodenum are slightly alkaline. The optimum pH is pH of 8 for the pancreatic lipase. This have been mentioned, I guess, in chapter 7. My video is still there, you can check it out. I'm gonna link it in the description box. Although changes in pH affect the activity of the enzyme, these effects are usually reversible. This is another difference between temperature and pH. Temperature have a permanent change, while in pH it is a reversible one. Example, an enzyme that is disabled by a low pH will restart its normal activity when its optimum temperature is met again. So here we use the graph to know which enzyme we're talking about. So for instance, pepsin have a pH optimum pH of 2, so that's why we know that this one is for pepsin. This one is at 7, which is for most enzymes and enzymes inside the cell. Now the one which have like an optimum pH of 8, here I'm trying to reach the 8, is the pancreatic lipase found in the duodenum. And that's all for today. Don't forget to subscribe for the channel to support me and see you in the next video.